Okay, so today we will discuss or start a new chapter. It is enzymes. So just give me a minute. Let me get ready. In this chapter, in enzymes, we will discuss properties of enzymes. Enzymes are globular proteins. They're globular proteins. And there are two types of enzymes. We'll discuss intracellular, extracellular enzymes. Then we will talk about mode of action of enzymes in terms of an active site, enzyme substrate complex, lowering of an activation energy, enzyme specificity. We will also discuss lock and key hypothesis and induced fit model. We will see how these two models differ from each other. What are the differences in between, lock and key model and induced fit model or hypothesis. Then we will discuss some investigations that involves progress of enzyme catalyzed reactions by measuring the formation of products and by disappearance of substrate. So this is another topic. We'll discuss the use of colorimeter in this chapter. And then we have factors affecting the rate of enzyme action. We'll discuss the different factors that affect the rate of an enzyme action. That includes temperature, pH, enzyme concentration, substrate concentration, and inhibitor concentration. And then we'll discuss Michaelis-Menten constant, which is the Km value, and we'll discuss immobilization of enzymes and a comparison in between the free enzymes and activity of free enzymes and immobilized enzymes. We'll also discuss the method of immobilization of enzymes. So these are topics in this chapter. Let's see properties of enzymes first, or an enzyme. Enzymes are globular proteins in nature. These are water-soluble proteins. And they have tertiary structure. Okay, so it means that they have a single polypeptide with a 3D shape, and tertiary structure, that involves 4 forces that we have discussed in the biological molecules: hydrogen bonding, ionic bonding, hydrophobic interactions, and disulfide linkages. We'll discuss the significance of these bonding in the shape or in the activity of enzymes. And enzymes are described as biocatalysts. Okay, so they are biocatalysts. A biocatalyst means a substance which speeds up the rate of an enzyme action without being involved in a chemical reaction itself. Okay, so biocatalysts can be described as, or enzymes can be described as, substances which speed up the rate of a chemical reaction without being changed at the end, without being changed at the end. Okay, so this is used as a description of an enzyme. We can use other properties of an enzymes, but there are many other substances that are globular proteins, like there are hormones that may be globular in. So these are not, the properties specific to enzymes. This is specific to enzymes only. Okay, and then enzymes lower down activation energy, lower down activation energy. We'll discuss what does an activation energy mean, and enzymes are specific to pH, temperature, and substrate. Okay, so let's see what does an activation energy mean. Activation energy. Okay, if you already know that every molecule or every substance that has some chemical potential energy, some substances have lower chemical potential energy, some substances have higher chemical potential energy. So if I plot a graph showing chemical potential energy on y-axis, this is chemical potential energy on y-axis, and if I plot time on, maybe x-axis, okay, and we compare the activation energy of a substrate, this may be a substrate, a substance like it may be glucose, okay, so this is a, or maybe sucrose, okay, so this is a substrate, and this substrate can be converted into glucose and fructose. So if we compare the chemical potential energy of this sucrose molecule, okay, with that of its products, which are, these are products, this is a substrate, we have this sucrose as a substrate, and we have these two products. So substrate has a higher chemical potential energy and product have lower chemical potential energy, okay. So technically this molecule sucrose should break down into these two molecules and should release energy, but sucrose does not convert into products at its own. It needs initially a higher energy to be provided, okay, so that this could be converted into the products at a lower energy, okay. So this is, as I said, initial or chemical potential energy of this substrate molecule, and this much energy is needed to start this reaction so that we can convert sucrose into its products, which is glucose and fructose. This initially, this amount of energy needed to start a chemical reaction is known as an activation energy. So this is an activation energy. Like in the last chapter in biological molecules, we have discussed that in this test for non-reducing sugars, that if sucrose is boiled with dilute hydrochloric acid, the sucrose will be broken down to glucose and fructose. So you see this boiling that provides energy, which is needed to convert this sucrose into the product. Okay, so this higher energy or this amount of energy needed to start a chemical reaction or to convert this product, this substrate into the product, is known as an activation energy. Okay. Now, what is the role of enzymes in these chemical reactions? This chemical reaction could be carried out at a boiling temperature, at a high temperature. Okay, in a food test for non-reducing sugar, we have already discussed, and you might remember that when we do a test for non-reducing sugar or for sucrose, we boil the sucrose sample with the dilute hydrochloric acid in a water bath, and this brings about hydrolysis of sucrose. Okay, but if we use enzymes instead of you know converting this substrate into the products without an enzyme, this is an activation energy without enzyme. But if we repeat this experiment and we want to break this sucrose into its products at a lower temperature, or maybe at a room temperature, we can use some enzymes. So if we mix this sucrose with a sucrase enzyme, we will not need this much higher energy to be provided to convert this sucrose into the product. Instead, there would be less energy needed to convert this substrate into the product. So new graph may be like this. So this is an activation energy. This is an activation energy. Activation energy with an enzyme. So this difference in this graph from this point to this point, from here to here, this shows decrease in the activation energy. Activation energy by use of enzymes. You got it? And this is the chemical potential energy of, the products, which is lower than the chemical potential energy of a substrate or that of sucrose. Okay. So by now we have discussed, the description of an enzyme. Enzymes are biocatalysts which speed up the rate of an enzyme action without being altered at the end of a reaction. And we have discussed the activation energy. Now let's see the model of an enzyme action, or mode of an enzyme action. An enzyme action. There has been two theories proposed to describe the mode of action of an enzyme. One is the lock and key theory or model, and the other is induced fit model. So let's see the lock and key model first. According to lock and key model, an enzyme has we can draw an enzyme in this shape. So according to lock and key model, enzyme has a 3D globular rigid fixed shape, and it has an active site. This is an active site which is specific or which is complementary to the shape of a substrate. So how do we describe this in active site? Active site is this cavity or this depression on one side of an enzyme where a substance can bind it in itself into. And if you look at this substrate is any substance that could be converted into the product by an enzyme. So if you look at this substrate molecule, this is, for example, represented with an S, which is a substrate, and we have this E, an enzyme. So if you look at the shapes of this substrate and this shape of an active site, they both are complementary shapes. Okay. And in a solution, these enzymes and substrate molecules are in a continuous motion due to the kinetic energy present in them. These molecules are constantly colliding with each other. And as a result of collision in between these two, it is likely that substrate molecules can bind into the active site of an enzyme. So let me just copy it again. Okay, so you see what happens if they are constantly colliding with each other. This substrate this binds into this active site of an enzyme, and this makes an enzyme substrate complex, ES complex. Okay, enzyme substrate complex. And this reaction is reversible. Okay, every substrate that binds into an active site of an enzyme may not necessarily be converted into product, and this may come out of the active site without being changed into the product. And it does also change the shape of an enzyme. Okay, so this is an enzyme substrate complex formed. There may be the hydrogen bonding formed in between the substrate and the active site of an enzyme molecule. So this is kind of hydrogen bonding that hold the substrate molecule into the active site of an enzyme. This enzyme now lowers down the activation energy, and it reorients, weakens the bonding in between the unit molecules of this substrate, and it converts this into, this substrate is converted into the product. Okay, so this substrate may be broken down into two parts, and it is converted into just give me a minute. So this part may be, this is converted into two molecules of products, and this enzyme molecule that remains unchanged. This is unchanged. So according to this hypothesis, substrate is like a key. Okay, substrate is like a key which fits into a specific enzyme as lock. So according to this lock and key hypothesis, the enzyme acts as a lock and substrate acts as a key. So as they are both specific to each other, to make an enzyme substrate complex, enzymes lowers the activation energy. It reorients the bonding in between the substrate molecules, and consequently, it weakens some bond that may be broken down, or it sometimes brings the molecules together so that the new bonds may be formed in between the smaller units to make the larger units. Okay. So this is lock and key theory or lock and key hypothesis. Now let's see an induced fit model and how an induced fit model that differs from a lock and key hypothesis. See, according to induced fit model, an enzyme does not have, a rigid shape. It is, though it is 3D and globular, it is 3D and globular. It has tertiary structure, but its structure is flexible. Okay, not rigid and not fixed. And its active site is also not complementary. Active site is not complementary to the substrate. Okay, so what does that mean? It means that if this is, for example, the same substrate as I have drawn earlier, so this is a substrate and this is an enzyme. So you can see the active site, this one part, which is an active site here. See, this is an active site. So this doesn't have a complementary shape to that of substrate. So what happens? A substrate binds with a substrate binds with an enzyme. Okay. So this is how this enzyme or this substrate binds with it. Okay. But still, you see, this shape is not complementary. Okay. So substrate bind with enzyme and this shape is not complementary to it. So it binds, substrate binds with an enzyme. And what happens then? It induces the change in the shape of an enzyme. Okay. It induces when it binds with it, it induces the change in the shape of an enzyme. And so that the shape of an enzyme is, okay, so that the shape of an enzyme is now complementary to the shape of a substrate. So you see there is a change induced in the enzyme active site by a substrate. And so that enzyme is exact fit into the, now it is complementary. Okay. Now it is an exact fit into the active site, and it is an enzyme substrate. So this is an enzyme substrate complex. And once this enzyme substrate complex is formed, what happens? The substrate is broken down to the product, and enzyme returns into its original shape again. Okay. So you see, this substrate is broken down to the products. These are the products formed. So substrate is converted into the product, and enzyme, if you look at this in enzyme returns to its original shape that was at the start of this chemical reaction. So enzyme is unchanged or unaltered at the end of this chemical reaction. So enzyme active site returns to the original shape as it was in the beginning. So that's why we say that the enzymes are unchanged at the end of a reaction, but during a chemical reaction, they can change their active site or the shape of an active site according to the induced fit model. So if we compare the induced fit model and lock and key model is has a rigid or fixed shape, okay, whereas this has a flexible shape, and the shape of an active site is complementary in case of lock and key model, whereas it is not complementary in case of induced fit model. And once the substrate binds with an enzyme, it induces a change in the shape of an active site so that the substrate has complementary or the enzyme has a complementary shape of an active site to the substrate, and substrate is exact fit into or precise fit into the active site of an enzyme. And after the substrate is converted into the product, the enzyme returns to its initial shape, so it is unchanged at the end of a chemical reaction. So that's about both these theories proposed to describe the model of an enzyme action. So let's see some questions on it, and then we'll move on to the next topic. Okay. Are you there?
Yes.
Great. So let's see, explain what is meant by an enzyme. Okay. So this question may be describe an enzyme, or it may be, and explain what is meant by an enzyme. As we have discussed many points to explain an enzyme. Enzymes, they are biological catalysts. Enzymes are biological molecules, or they are proteins in nature, and they catalyze or speed up the chemical reactions. So they increase the rate or the speed of a metabolic reaction or that of a chemical reaction that takes place in the cells, or the biological or cellular reactions. Or you can say these are globular proteins, and they remain unchanged at the end of a reaction. Okay, they are not used up during, so they can be reused, and they lower down an activation energy. So you can write two of these points, but there are many points that could be used to describe and explain this enzyme. As we discussed earlier, there are two types of enzymes. There are extracellular, means enzymes that work outside the cells, and there are intracellular that work inside the cell. So you should know some examples of the enzymes that work inside the cell or that work outside the cell. So this is an example. In human tears and saliva, lysozyme acts as an extracellular enzyme. State what is meant by the term extracellular. Means they act or they catalyze the chemical reactions outside the cells. Catalyze chemical reactions outside the cells. Okay. Or if there are intracellular enzymes, intracellular enzymes, they catalyze chemical reactions inside the cell. Inside the cell. Okay. For example, the enzymes involved in DNA synthesis or RNA synthesis that obviously take place inside the cell or inside the nucleus. So DNA polymerases, RNA polymerases, they are all intracellular enzymes. Whereas digestive enzymes may be intracellular, they may be extracellular, like proteases, lipases, carbohydrases, amylases. They may work inside the cell or they may work outside the cell. So they may be extracellular, they may be intracellular. A disaccharide trehalose is a reserve store of energy for this kind of a organism, S. cerevisiae, Saccharomyces. This is kind of a yeast. When glycogen stores decrease, the monomer of glycogen and trehalose is an alpha glucose. We have discussed structure of glycogen in the biological molecules, but the structure of trehalose is not in our syllabus. That information has been given in the question. The hydrolysis of trehalose is catalyzed by two different enzymes produced by this, regulatory trehalose and non-regulatory trehalose. So let's see what is question on it. Regulatory trehalose is found only in the cytosol. Cytosol means the cytoplasm of the cell, which is the fluid part of the cytoplasm, and non-regulatory trehalose has been found on the external surfaces of the cell, surface membranes, and inside the cell. State the location inside the cell where non-regulatory trehalase is likely to be found and explain the reason for your answer. Okay. So if you have understood this question, regulatory trehalose is found in the cytosol, the fluid part of the cytoplasm. And what is its role? We will have to look at its role, okay, to understand where it could be found. This is involved in the hydrolysis of trehalose is catalyzed by two different enzymes. This causes hydrolysis. And you know, hydrolytic enzymes are found where? Hydrolytic enzymes are found in where? The lysosomes of a cell. And that's why the lysosomes are involved in, phagocytosis and removal of the damaged or dead cell organelles, and even autolysis or the removal of the dead cells from the body. So that these are the lysosomes, cell organelles which contain hydrolytic enzymes in them. So hydrolysis takes place in these lysosomes that we have discussed in cell structure. So that must be state the location inside the cell. So you should write lysosomes. They are found in the lysosomes. And explain the reason for your answer. What is the reason for this answer? And it is for 2 marks. So you should give reason. The reason may be that they have suitable conditions for their activity inside the lysosomes. Okay. Or the reason may be they are enclosed in the cell membrane. So they only are used for the hydrolysis when they are needed. Otherwise, if they are freely present in the cytoplasm, they would bring about hydrolysis of the substrate molecule if it is found in the cytoplasm. So lysosomes contain these enzymes as they have suitable pH for their activity. Okay, so that maybe you can write about the suitable pH, okay, or specific pH, or maybe you can specify some acidic pH, or maybe you can specify some low pH, okay. Yes, and explain whether both types of trehalase, this is an enzyme, regulatory and non-regulatory, can be described as intracellular enzymes. Intracellular enzymes are which work inside the cell. So, one type that is found in lysosomes, obviously this is an intracellular, but this question describes that the other kind, the other kind is present on the inner surface. This regulatory is present in the cytosol and the lysosomes, and non-regulatory has been found in the external surfaces of the cell surface membrane and inside the cell. So both enzymes can work inside, but non-regulatory can also work on outside of the cell. So non-regulatory can be an extracellular and an intracellular, but this regulatory is intracellular only. So yes, you can say that which works inside the cell. So both work inside. So yes, they are intracellular. Okay. Now let's see, explain the mode of action of enzymes in terms of an active site. Enzyme substrate complex, lowering of an activation energy, enzyme specificity, including the lock and key hypothesis and induced fit model. You have question on it. Elastase has an active site with a specific shape. The mode of action of this enzyme supports the lock and key hypothesis. Explain the mode of action of elastase. Like you are asked to describe the lock and key model. Okay, and you may use the space below to draw a diagram or diagrams to help your answer. So you can draw a diagram here and you can annotate it. Or even if you don't draw a diagram, that doesn't matter. Why? Because this is just 3 marks question, and you should not spend a lot of time on answering this question. Okay. So most of the time, students finding such questions easier and having a lot of information on it, they spend a lot of time on such answers, and then at the end they lack time to answer some questions. Okay, so the first thing that you should do is you should know what number or what marks are there for answering this question. So there are 3 marks, so you should write 3 points, and you should think clearly and make a rubric what 3 important points should be there. Okay. Okay. So you should think about is that the shape of an active site complementary or not complementary? It is complementary. So would that substrate fit into active site? Yes, it would fit into an active site, and this would make an enzyme substrate complex. And what would happen if an enzyme substrate complex is formed? You see, activation energy is lowered, and then this would convert a substrate into the product. Okay, so these may be the things, right? So let's see. There are many points given in it. The first point is shape of substrate or elastin complemented the shape of an active site. Okay, so in a lock and key model, the shape is complementary, so that's great. You should avoid writing this term matching or the same. This is not matching or this is not same. R stand for reject in this case. Or description substrate fits into an active site. Obviously, that fits into an active site. You can, on the diagram, you can label this. Or if you don't have the diagram drawn, that's fine. Now, lock is an enzyme and key is a substrate. And they join together or they bind together and they make an enzyme substrate complex or ESC. And this happens why? Because enzyme and substrate are in continuous random motion and they collide with each other. Higher is the random motion, more will be chances of substrate molecule to bind into the active site of an enzyme. Okay. So then what happens? Peptide fragments, peptide products released from or except amino acid. So you see, the substrate is converted into the product. This is what you can write here. And then following points need to be annotated. If you have drawn a diagram, you should annotate your diagram. Hydrogen bonding or temporary bonds between enzyme and a substrate. And you can write about activation energy. This EA is lowered. EA stands for activation energy. Okay. Or you can write that enzyme puts some strain on the peptide bond, or there is some kind of an electron transfer brought about by an enzyme, and the reactants held together for bond forming. That may be or bond breaking. Okay. So there are many points that there are 6 points, and out of these 6, you can write any 3 of them. And try writing the short statements instead of using the conjunctive words, and instead of writing the longer phrases, you should use short sentences. Okay, short statements, or when you're answering such questions, and avoid joining words, or what are joining words known as conjunctions, probably in English. Okay, so avoid using 2 statements together by conjunction, so that this confuses the concept and makes it difficult for an examiner to answer fully. Let's see then. Lysozyme uses the induced fit model. Okay. The earlier question was on lock and key model, and this is on induced fit model. Explain the mode of action of an enzyme that uses induced fit model. So simply what will happen? Substrate shape not exactly complementary to the active site. So this is the first important point. Lock and key model में हमने discuss किया था कि in lock and key model, the shape of the substrate is exactly complementary or is complementary to the active site, whereas in induced fit model, it is not complementary. Or shape of an active site is flexible. Okay, so this is flexible. Slightly it can change. Whereas in case of lock and key model, it is rigid. Okay, if you compare it with lock and key model, it is not, it is complementary. The shape is complementary. And active site is rigid instead of flexible in the induced fit model. And once the enzyme and substrate bind together, the substrate induces a change in an active site so that now it becomes complementary and it is a better fit. It makes an enzyme substrate complex, and then again, once the enzyme substrate complex is formed, the substrate is converted into the product by an enzyme by lowering down an activation energy. So the products are formed. As the products are formed are no longer complementary to the shape of an active site, so they come out of an active site. So there are 4 points. Okay. So this is another question. Figure 4.1 shows 2 ways in which enzyme interact with their substrate. So this is an enzyme A and this is an enzyme B. So you see this binds into it and this makes an ES complex, and this is also an ES complex. Okay. So explain the difference between the 2 ways in which enzyme interact with this substrate as shown in figure 4.1. So look at the shape. Do you see initially these 2 shapes are complementary to each other? If you know, this shape is removed from here, and if we fix it into it, is it an exact fit? Yes, this is an exact fit. So this is lock and key. This is lock and key model. But in this case, if you look at this, is this an exact fit? No, this is not an exact fit. So this has induced a change in this shape so that this is now an exact fit. So this is an induced fit model. Induced fit model. So this is what you have to do in this case. The question begins with an explain the difference between the two. So you have to give reason why, you know, this is an exact fit here and this is, you know, initially not an exact fit, but then becomes an exact fit. So do you see enzyme A uses lock and key model, whereas enzyme B uses induced fit model? Why enzyme works by lock and key model or this by induced fit model? Or enzyme A lock and key, shape of active site is complementary, and enzyme B induced fit model has an active site that molds around the substrate or is flexible. You can write it is flexible, and it has been, a change has been induced in it. So you can write in alternative words. This is what does AW means, you know, alternative words. So you do not necessarily need to write these words. You can write your own words as we have discussed earlier. Look at this now, an activation energy. The graph shows energy changes in a chemical reaction. We have progress of a reaction or a time on x-axis, and we have energy level on y-axis. What is the activation energy when an enzyme is added? Okay, so this graph shows without an enzyme. Why? Because this have a higher activation energy. And this graph shows activation energy with an enzyme. So activation energy should be recorded from here, from this level of this substrate. And this shows an activation energy with an enzyme. And this shows an activation energy without an enzyme. Okay. So the question is, when an enzyme is added, so 2 should be an activation energy with an enzyme. What is this 1? 1 is this decrease in activation.
Yes.
Activation energy. Okay. And what is 3? We don't need to, you know about this 3 and this 4 actually. Okay, so 1 is the decrease and 2 is the activation energy, or this could have been, activation energy without an enzyme. So this is B is the correct choice in this case. Now let's see, we have the similar question here again. The graph shows the effect of an enzyme on a reaction. We have a time on x-axis and we have a energy level of a substrate, energy level of a product here. Which labels are correct? And this is decrease in energy level, and what else is there? X is the reaction without an enzyme. X is reaction without an enzyme. X is this one, dotted line. Yes, this part is correct. W is the activation energy. No, W is not an activation energy. Activation energy should start from here. This should be an activation energy. Okay, so this second part of the statement is not correct. First part is correct, but the second part is not correct. X is the reaction with the enzyme. X is the reaction with the enzyme. No, X is not a reaction with the enzyme. This is without an enzyme. So this first part is not correct. Y is the reaction with the enzyme. Y is the reaction with the enzyme. Yes, this is correct. First part is correct. And W is the difference in an activation energy. Yes, this is difference in an activation energies. The second part is also correct. So C is a correct choice. And we should look for any error in the D as well, so that we can reject this D. Z is energy gained by a product. No, product has not gained this energy. Okay, this is the energy that a substrate has lost to turn into a product. And W is the activation energy with enzymes. No, W is not an activation energy with enzyme. Okay, activation energy with the enzyme is here, number 1, and activation energy without an enzyme is this, number 2. And this is W is the difference, or you can say the decrease. So explain how enzymes lower the activation energy needed to allow reactions to produce. We discussed that enzyme substrate complexes are formed, and when enzyme substrate complexes are formed, for example, this is an enzyme and this is a substrate. Okay, and there is some kind of bonding formed in between this, so that holds this substrate into an active site. It is hydrogen bonding generally, and as a consequence, this binds here. It puts some kind of strain here at this molecule. Okay, so this is a strain on the substrate, so that here the bond may be broken down and this substrate is converted into 2 products. So this may be a way, a substrate is converted into the product. So this is what we have to write in this case. How does it lower down the activation energy? Provides alternative pathway, brings reactants close together in some cases, and puts a strain on the reactants, and so bonds break more easily, and transfer of charges or groups, or AVP, that stands for alternative valid point, or involvement of R groups. You can write involvement of R groups means the R groups of this active site and this substrate can make a kind of bonding here in between them, so that they could hold a substrate molecule into the active site of an enzyme to make an enzyme substrate complex. Hope you have understood it.
Yes, sir. Okay.
So that's it for today. In the next class, we'll discuss rate of an enzyme action and the factors affecting the rate of an enzyme action.
Okay, sir.
Okay, thank you.
Okay, sir. Allah Hafiz, and thank you so much.